So let me go back and see if I can pick up a little bit of this. All right, so ribosomes have two parts. So just ignore kind of what I'm saying because I forgot to start recording. So we got the top part is the big part, the bottom part. What did we say the bottom part looked like?
A snowboard.
Thank you. So we've got a dinosaur riding a snowboard, whatever you want to remember it.
Thank you, Courtney.
And we are making proteins. That's what ribosomes do using amino acids. Now, so let's take a look at this picture. This is a real picture. This is a TEM. Any ideas what that means?
Transmission electron microscope.
Thank you! All right, so transmission electron micrograph. Okay, so this is using electron microscopes. Really, really small ribosomes. And what we're actually seeing is the ribosome making the protein. So this little thread thing snaking out is the actual protein. And they're calling it a polypeptide chain. But polypeptide just means protein. Okay. So what you're actually seeing a view of proteins being constructed out of this ribosome. Something else to put in your notes. Ribosomes are found kind of scattered. So they're located in the cytoplasm, just kind of floating around, or attached to something called the rough ER. Who has my ER? Brandon does. Okay. So rough ER has these ribosomes embedded in it, kind of stuck in it. Yeah, Courtney.
You see that the C word, this cytoplasm?
Yeah.
It's a Y. Is that a Y?
Where?
It looks like a U and then an ampersand. Now what is it?
Cytoplasm.
Wait, a U? It's a U?
No, it's
Y.
It's not cutoplasm. It's cytoplasm.
Oh, I said pseudoplasm. Yeah.
So you gotta listen. Sometimes you can't trust the writing. Okay, you have to listen. I say everything I write anyway.
Is that R-E-R?
Yes. What does the R stand for?
Rough.
Rough. Rough ER. And we're going to talk about that when we get to Brandon. Okay. Now, I wanted to show you, because it's kind of hard. Boom. Okay. So hopefully you got a better idea now. You've got ribosomes building the proteins, and if, I don't know, can you see? Look right about here. Do you see in this zone? Do you see a little line?
Yeah.
Okay, that's that RNA that came out of the nucleus and it's being decoded. Okay, and man, just in one sequence, you might have hundreds of these ribosomes just going down the line, making proteins. Proteins are just spewing out the end. Okay. Questions on ribosome? Woo! All right, next one.
Woo!
Brandon, tell us a couple things about it.
The rough ER has to do with the proteins.
Okay.
And then the smooth one is like steroids and stuff.
All right, so let's get function first. We'll tackle the picture in a second. So there's two kinds. What Brandon was just talking about, there's rough ER. I'm not going to spell ER out all the time, okay? But that's what it stands for. Endoplasmic reticulum. Okay, big fancy word. Rough ER, notice what it has. It's got all these ribosomes. And what do ribosomes make? Proteins. So these are mostly used for transporting proteins. It's like a maze. It's like a series of channels that when these proteins get made, they're going to be navigating through this maze to get to different parts of the cell. All right, this is an extensive network of tubes that allow materials to travel through the cell. Okay, so if I'm a protein and I just got made, whoop, I could use this to travel a variety of different places. And this is three-dimensional, and we've sliced it in half. Okay. All right, what was the other part? Smooth?
Smooth ER.
What does it do?
It is with steroids.
Transports steroids, fats, other lipids. If you think back to our lipids category, it's the fats, it's the oils, it's the steroids, it's the waxes. And I think an easy way to remember that smooth transports those is think about grease and oil, and you get it on your hands, and
Wait,
it's
are we talking about the smooth
the
stuff right
smooth,
now?
the smooth ER. Okay, the smooth ER still transports, but it's transporting these things, not protein. Okay. Another difference. Look at the two besides the color. Rough has the ribosomes, smooth doesn't. Okay. So I kind of mentally think if I were to feel this somehow, it would feel bumpy, rough, and this would feel smooth. Okay. Either way, however you want to remember it, but they're transporting different things throughout the cell. All right. Anything else, Brandon? Those are the key points.
It's like located in the membrane.
Yeah, I mean it's located, and it doesn't show the whole thing here, but it connects to the nucleus, and it goes all the way to the cell membrane. Okay, so it covers a vast amount of the cell. That might be worth writing down too. Covers a large part of the cell. Now, for the picture, good luck.
Okay.
Okay.
Can you bring that?
Couple things I would try. First of all, I draw the nucleus as kind of a ellipse, some sort of circle. And then depending on how intricate you want to get, you can then do kind of a maze. Or what somebody yesterday did, after drawing the nucleus, they just kind of did this back and forth, kind of the series of channels, and then kind of interconnected some of them to make it look more maze-like. Okay, if you want to get fancier than that, go for it.
Ta-da.
Okay, rough. Rough has ribosomes. Smooth doesn't. Okay, and the smooth kind of looks more tubish, I think. Less maze-like and more tube-like. Macaroni.
That sounds good.
Okay.
I'm having that for supper tonight.
And there's no ribosomes. Don't get carried away on the ribosomes. Smooth doesn't have any ribosomes. Okay.
Wait, I'm not done.
That's fine. I'm just going to move it slightly a little bit. Okay. Now, obviously this is an artistic rendering. That's not what it really looks like. Okay. Down here below, we've got a TEM, another transmission electron micrograph. Are we done with this picture?
Yes.
Okay. Let me slide it down. Okay, check that one out. If you had to take a guess, smooth or rough, what's your clues?
The nucleus.
Well, I don't see the nucleus actually in this picture.
Oh crap, I thought the dots.
The dots. What are the dots? The ribosomes. Look at all the dots. Holy smokes. Okay. So what you're actually seeing is a cross section. It's almost like we've just cut the top off. We're looking down on this, and I'm seeing this maze of networked channels. Okay. And I'm seeing lots and lots of ribosomes. Rough, ER. All right, let's compare that to smooth. Whoa. From here to here is smooth ER. What's different?
Scattered.
Man, it's all scattered, isn't it? What other differences?
Not as many dots.
Not as many dots. Actually, I don't see many at all. Now this CJ is something else. Okay. Actually, to be truthful, I'm not quite sure what it is. Okay. Ribosomes would be a lot smaller though. Ribosomes would be like that size compared to whatever that is. Okay.
What are those dots in mitochondria?
Where?
Are
So
those?
these are mitochondria. Those that kind of look the same.
Yeah,
Now
but
this is rough ER.
Yeah.
Okay, because you can see all those little dots on the outsides of those. Okay. In terms of transport, if you look at the rough ER, to me, that looks organized. Somebody yesterday said, that kind of looks like a fingerprint. Yeah, I can see that. I can see that. And if I'm a protein and I'm here and I need to get to over here, boom, it's a straight shot. That's like interstate. If I'm a steroid and I'm here and I need to get here, am I going to get a straight shot?
No.
No. Okay, I might. Boop, 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 boop. That's not too efficient. Okay, smooth is like back roads that might not actually go where you want them to go. You get lost doing GPS. Okay, the top rough ER much faster. Okay, because proteins are very, very critical to a cell's life, and the proteins have to get where they need to go very quick. So that's another big difference between these two. Okay, questions on ER? Golgi. Let's see, who is my Golgi
That's
expert?
how you say it.
Yeah, Golgi
I thought it was
or Golgi, if you want to throw in an extra G, but Golgi. It's like a J. Yeah, it's like Golgi. All right, now nobody actually claimed this one, so I'll cover it. Want to do picture first or function?
Picture.
Okay, let's look at the picture.
Looks like a mud. It looks like upside down mud.
Yeah, okay.
Looks like someone just
Been
stepped in
splattered
mud.
by a
Splattered.
All right, first thing you should notice is it kind of looks like ER.
Like what?
Does it kind of look like the ER?
Yeah.
Kind of the maze stuff. The difference here is when you draw it, they don't interconnect. So you got a stack, and then you've got another one, and then you've got another one, but they're not connected to each other. Okay, somebody, actually my high school biology teacher called it a stack of pancakes. Okay, you could have 5, 6 pancakes all in a stack. They're touching, but they're not actually connected to each other. Okay. These stacks are called, if you can't read the word from where you are, cisterns or cisternae. All right, it's a Latin phrase. Cisternae means sack. That is something you should probably put on your diagram. Okay, make sure you know what that is. A cisternae are these sacks, these flattened sacks. Okay. And they're anywhere, I don't know, I think somebody yesterday, somebody who researched it said there's anywhere between 5 to 8. It kind of ranges, but there's several of these kind of stacked on top of each other. What do you notice about the size of the cistern? As we go from top to bottom, what happens to the size? They get bigger, and then what? Okay, and they kind of look like they're falling apart on this side. Good observation, Brandon. The way this works, and whoever's got vesicles, did anybody have vesicles in here? Yeah, Wade and Austin McVeigh did. Vesicles, which is the next one down, so we're going to do both of these at the same time, ladies. Make sure you're catching this. All right, so vesicles, which is right below Golgi, this is storage sacs. Okay, it's like a container. They're called vesicles. You'll see newly formed vesicles. You've got incoming transport vesicles. And all that really looks like is just it's a sphere. It's got some sort of cargo inside of it, but it's kind of like a package with some sort of material on the inside. Okay. Well, sacks in this form. Yeah, it's not like a grocery sack. S-A-C-K. Okay. Let's compare it to a real picture. Do you see it? You see the layers? Okay. Now, one thing that's kind of cool about the Golgi is it receives materials. This has to do with the function. It's receives material. And that's why the top is kind of fat, because it's receiving materials. And then as it goes through these different layers, it gets changed. It's mostly receiving proteins, and it will modify those and package them. So it's kind of like a UPS, some people have called it, or a post office. That's pretty good. If I'm wanting to deliver something to Grandma for Christmas, I'm going to take it to the post office, or I'm going to have UPS ship it for me, FedEx, and they're going to put some labels on it. They're going to modify it by putting it in some sort of package that is good to ship, and then they're going to send it to a destination. So this side, the trans face, these are all things that have been packaged up and are being sent someplace. And not all of them are going to the same spot. Some are going here. This might be going that direction. These are going over here. Somehow the cell knows where to send it. Pretty cool. Okay, let me show you a little animation of this one. Okay, let's do a couple more. Let's take a look at this one. Who is my mitochondria expert?
Yes.
Austin Moore. Okay, tell me a couple functions about it. What's it do?
It creates ATP.
Yeah.
Energy.
Yeah. Which is cell energy. Do you remember how it does this? What is it breaking apart? I'll give you some help. Okay. All right, so everything that you eat at some point will get absorbed into your cells chemically. Okay, your breakfast, your lunch, your dinner gets broken down physically and then eventually chemically into smaller and smaller pieces. Okay. We already did vesicles.
Oh.
Yes, we did. That's why I said you have to make sure you're paying attention.
I think I put
It's,
that on.
it's kind of part of Golgi. They're kind of related. Vesicles and Golgi. The vesicles are the sacks, those containers, the boxes.
Oh.
Okay, they're just containers. So when you draw the picture of a vesicle, it's just a sphere or some sort of round blob. Okay, it's just a container.
And it packages.
It's just a packaged material. Okay, it's like if I go to the grocery store and buy some groceries, I'm going to put it in a container, but it's just for transporting. I'm not going to leave it in that. I'm going to eventually put it in a pantry or something. Okay, so the vesicle in your cell, it's just a container. It's a temporary holding place. All right, mitochondria creates ATP, which we really haven't talked about too much, but this is your cell's battery. It's your cell's energy molecule. Okay, and the way it does this is it breaks apart food chemically. So if I've got a molecule of glucose that goes into this thing, what's going to come out is this energy molecule so that our cells can do what they need to using the energy that it's producing. So it kind of has, what's kind of just a nickname or something that...
The power plant.
Thank you very much, sir. Think of it like a power plant. All right, your power plant over in Richmond. Coal goes in, all right, it heats up water, drives turbines, that generates the electricity. Okay, the coal doesn't stay in there, all right, it's converted. The energy in the coal is converted to electricity, all right, the electrical energy. So that's kind of what your mitochondria do. What kind of cells might need a lot of these?
Muscle cells.
Yeah, muscle cells have thousands. Okay, whereas skin cells might only have a dozen or so. But your muscles require tons of energy, and so they need lots of these little microscopic power plants. Okay. All right, picture time. Well, there's two things that you should notice. It has an outer membrane that is smooth. All right, think of it kind of like a pill or a capsule. Okay, it has this nice smooth exterior. And what we've done here is we've done a little slice and cut a chunk out. Okay? That's the outer membrane. The inner membrane has all these folds. So when you draw this one, you kind of have to do this back, forth, back, forth, back, forth, back, forth. Okay, back, forth. And that basically creates a lot of surface area. Increased surface area. So there's a lot of chemical reactions that can happen inside a very, very, very small space. And those are called the cristae. I'll give you a fancy scientific name. The cristae are these folds of the inner membrane. Can you see them on the real picture? Kind of. Okay. So right here, all these little things, that's the inner membrane, that's the cristae. And I don't know, I always think the mitochondria looks a little bit like a potato. Okay.
Danger.
That's bad. Oh wait, I better check.
Someone just went, ow. That's so funny. That actually hurt. Sound like
That's
sorry.
good. Just double checking.
I'd probably record that.
See?
They broke the art display over there.
Oh.
Yeah, they were putting something in and it slipped. What's that? Oh, come on.
That is a mitochondria.
No, that's the mitochondria.
VR.
What's this? What kind of VR?
Rough.
Rough VR thinking, Brandon. Okay, question on mitochondria, you got your picture. We'll do a couple more. Lysosome. Who's got that one?
That was like an egg.
Ryan, shoot, nobody's here. Okay. Picture on this one's super easy. Draw looks like a marble. Put some dots inside. Or you can draw it like this. Have you ever had a pomegranate?
Yeah.
It kind of looks like a pomegranate. Has all these seeds and stuff on the inside. Okay. Okay, so that one's pretty easy to draw. This is a type of vesicle. So a container, a sac. It's a type of vesicles. And the difference is what they're carrying is dangerous. These yellow, orange dots are digestive enzymes, which is another word for protein. These are powerful chemicals that if they contact something, destroy it, digest it.
Is that what happened to the one?
Yeah, so in that previous animation, the Golgi, that one vesicle which kind of hung out, a bacteria came in, it engulfed it, and then kablooey. All right, it destroyed it, it digested it. Sometimes these will destroy, well, destroy is not a good word because it implies that the matter is gone. It recycles things, okay, but it destroys worn out organelles. So let's say a mitochondria's outlived its usefulness. The cell will send some lysosomes to merge with it and break it up into tiny pieces, and it could be recycled. Sometimes these can destroy the whole cell. I'm sorry?
You made the smallest box have the most right.
Ah, I'm sorry. Right smaller. All right, so it can destroy the whole cell even. So sometimes these are called suicide sacs. Because if they open up inside the cell, then basically their contents just spill out on the whole cell and can destroy the entire cell. There's only a few cases when that should actually happen. And a good example is your hand.
Your hand?
Your hand. Okay, you all have separate fingers.
Yes.
At one point in your life, you didn't. You had a paddle.
No way.
Okay. Now this is really early. All right. This is like maybe a month after conception. Okay, but month and a half when you were still in the womb, if we were to look at your hand, it looked kind of like a paddle, a duck web. Okay. And what happened is the cells located between your fingers were programmed to self-destruct. And when those self-destructed, you started to get separate fingers. But then there's other cells that didn't do that. Otherwise, you would have just had no hand whatsoever.
Does that happen or something?
Sometimes there are some cases where
The
the
cell
process
doesn't
doesn't finish, and so the palm actually extends up farther to like the first knuckle.
Whoa. Did they take that out?
They can surgically. I mean, it's pretty easy to just cut it and then hold it down. Okay, so it's not that critical of an issue, but in animals that have webbed feet, the cells that are connecting the fingers just don't self-destruct. And the programming is different, the DNA is different. But that's a good example. If you've ever had a cast on an arm or a leg for a long time, I don't know, has anybody ever had that?
No.
I don't wish it upon you, but if you did, when they took the cast off, what did the muscle look
It was like really blue.
like? It was a lot, yeah, it was a lot smaller. The only time I've ever broken something, I broke my wrist at Disney World when I was really little. I tripped on my shoelace. Okay, that, you know, you don't think that ever happens, but I was pushing my younger brother in a stroller. I tripped on my shoelace. I put out my hands to catch myself, and that's actually one of the easiest ways to break your wrist.
Oh, trust me.
Well, I know, yeah, so yeah, you're not supposed to put your wrist out and just crash to the floor, but when you put your hands down, you have a compression fracture because all that force gets shoved onto your wrist and something's going to give. So they had I had up to the elbow for, I don't know, probably about a month or so. And then when they took it off, I mean, my forearm was like gone. I mean, there's just no muscle whatsoever. And it's because some of my muscle cells had atrophied. The lysosomes had actually been programmed to, well, we're not using this stuff. Let's get rid of it. Let's recycle it. Now you can build that back up, but it does take a little while. All right, let me show you this animation, and then we'll get a little. All right, let's get our last one for today. Cytoskeleton.
Okay, I need that. I can do that.
Let's look at notes. Let's do the notes and function. Okay, first thing I would put down in that box are these three words, and try to maybe space them out just a little bit. Okay, in the notes and function section, there's three parts of the cytoskeleton. We've got microtubules, microfilaments, and intermediate filaments. We're not going to worry about the picture just yet, because it gets a little nuts. Microtubules, keyword there is tubule. All right, this is actually a tube. These are used for movement. It could be the whole cell moving. We'll talk about cilia and flagella next time. It could be used to move things inside the cell, which is really bizarre. Okay, we can move things inside the cell using something called a motor protein that'll actually walk. It looks really bizarre. It walks on these tubes, okay, like a channel. Microfilaments are a lot smaller. These are used for shape. They define the shape of the cell. Not all cells look the same. In the human body, we've got over 200 different types of cells. Each one has a distinctive look, different shape. And it's mostly due to those microfilaments. The last part of the cytoskeleton are called intermediate filaments. These organize the cytoplasm. It's kind of like a scaffold. Right? There's all these connecting, intercrossing fibers that things can attach to. And it also just helps keep things where they're supposed to be. So if we look at our picture, I don't know how you want to draw it. It's up to you. You can draw 3 separate pictures. That might work. Microtubules are these right here. They're the biggest ones. They're these big tubes that run parallel to each other, and they'll usually go the length of the cell, and they're just literally a tube of protein. So if you want to just draw a more, okay, but they're the biggest ones. They're these long, big diameter tubes. The microfilaments, little guys. Okay, compared to this, it might look just like a thread. Okay, and they're really small, but they're attached to the surface. Okay, usually right on the inside of the cell membrane are those guys, right on the inside. And then the last ones, the intermediate filaments, are these green, bungee-looking cord things. Okay, and you'll notice they kind of look like a weird spider web that are kind of just, they're attached to the cell membrane, but they're attached to the mitochondria, they're attached to the ER, they're attached to the microtubules, they're attached. They're attached to all sorts of stuff. And that's what helps kind of hold everything in a spot. Now the real picture looks like that.
How am I going to draw that?
All right, now if you had to take a guess, okay, these are different sizes. Okay, this is like microfilaments. These are more like intermediate filaments, and that kind of gives you some idea of size difference. Okay. All right, questions on any of that?